If you want to understand why fasting protocols that were dismissed as pseudoscience for decades are now being studied at the world's most prestigious cancer research centers, including Johns Hopkins, MD Anderson, and the Mayo Clinic, don't look at calorie restriction theories. Don't look at weight loss mechanisms. Look at what happens inside cancer cells versus normal cells when nutrient availability drops for just 5 consecutive days. One specific eating protocol costs nothing, requires no special foods or supplements, triggers biological mechanisms that selectively starve cancer cells while simultaneously forcing your body to activate dormant stem cell populations and rebuild damaged immune systems from the ground up. Here's a number that should absolutely stop you cold. Researchers at the University of Southern California demonstrated that a 5-day fasting mimicking protocol reduced chemotherapy side effects by up to 70%, while simultaneously making cancer cells more vulnerable to treatment and protecting normal cells from damage. The same intervention that starved cancer cells provided protective effects for healthy tissue. This is the opposite of how we typically think about cancer treatment, where therapies kill cancer cells but also devastate healthy cells. Here, we have an intervention that harms cancer while protecting normal tissue through exploiting fundamental metabolic differences between the two cell types. Hi, I'm Doctor. I'm an emergency medicine physician and metabolic oncology researcher. And over nearly 16 years practicing emergency medicine, I have watched what happens when cancer reaches advanced stages. I've seen the devastating side effects of chemotherapy and radiation. I've watched patients become so weak from treatment that they can barely function. And I've also seen emerging research over the last decade that suggests we've been approaching cancer completely backwards. Instead of just poisoning cancer cells and accepting collateral damage to healthy tissue, we should be exploiting cancer cells' metabolic vulnerabilities through interventions that simultaneously weaken cancer and strengthen normal cells. In this video, I'm going to show you exactly what happens during a 5-day fasting mimicking protocol at the cellular level in both cancer cells and normal stem cells. Why this intervention triggers a biological reset that researchers are calling differential stress resistance, where normal cells become protected while cancer cells become vulnerable, and the exact 5-day protocol based on clinical research that has been tested in human cancer patients with measurable results. Most people have been told that fasting is dangerous, that you need to eat constantly to maintain energy and health, that missing meals will slow your metabolism and cause muscle loss, and that anyone with serious illness should definitely not fast because they need all the nutrition they can get. You've probably heard warnings about starvation mode, metabolic damage, and nutrient deficiency from prolonged periods without food. But after spending 2 years deep inside the literature on fasting, autophagy, and cancer metabolism, I've come to understand something that completely contradicts conventional nutritional advice. Short-term fasting, particularly in the 3 to 5 day range, triggers profound biological adaptations that have nothing to do with starvation and everything to do with cellular optimization, repair, and renewal. These adaptations are ancient survival mechanisms built into human biology over millions of years of evolution. And they create conditions that are simultaneously protective for normal cells and lethal for cancer cells. Think of your body like a complex ecosystem that operates in different modes depending on resource availability. In fed mode, when nutrients are constantly available, your body prioritizes growth, storage, and rapid energy utilization. Cell division is active. Protein synthesis is high. Growth signaling pathways are turned on. This mode is optimal when you're young, healthy, and need to build and maintain tissue. But this mode also supports cancer cell growth because cancer cells hijack the same growth pathways that normal cells use. In fasted mode, when nutrients become scarce, your body switches to a completely different operating system. Growth pathways shut down. Cellular repair and recycling mechanisms activate. Stored energy gets mobilized. Stem cell populations enter protective states, and your immune system gets rebuilt from scratch. This mode is optimal for cellular cleanup, damage repair, and system optimization. And critically, cancer cells cannot adapt to this mode effectively because their metabolism is fundamentally broken. Now here's where the differential stress resistance mechanism becomes crucial. Normal cells respond to nutrient deprivation by entering a protected low metabolism state. They shut down unnecessary processes. They activate stress resistance pathways. They focus resources on survival and essential functions. It's like a normal cell battening down the hatches during a storm, going into emergency conservation mode until conditions improve. Cancer cells cannot do this effectively. Cancer cells are locked in growth mode due to the oncogenic mutations that made them cancerous in the first place. They cannot downregulate growth signaling. They cannot enter protective quiescent states. They're essentially stuck with their accelerator pressed to the floor. So when nutrients disappear, normal cells slow down and survive, but cancer cells keep trying to grow and divide without adequate resources, which creates catastrophic metabolic stress that makes them vulnerable to death. The researcher who pioneered this concept is Dr. Walter Longo at the University of Southern California. His laboratory has published over 15 years of research demonstrating this differential stress resistance mechanism in multiple cancer types. Let me walk you through the landmark studies because the evidence here is not theoretical. It's concrete, measurable, and has been replicated across multiple institutions. In an early study published in the Proceedings of the National Academy of Sciences, Longo's team injected mice with aggressive cancer cells, then divided them into groups. Some mice were fed normally. Others underwent 48 to 60 hours of complete fasting before receiving chemotherapy. The fasted mice showed dramatic protection from chemotherapy toxicity. Their white blood cell counts, which typically plummet with chemo causing dangerous immunosuppression, remained significantly higher. Their red blood cell count stayed stable. And most remarkably, 8 different chemotherapy drugs tested showed this protective effect. The fasting didn't reduce chemotherapy's anti-cancer activity. It selectively protected normal cells while leaving cancer cells vulnerable. But the truly groundbreaking finding came when they tested fasting alone, without any chemotherapy. Mice with metastatic cancer underwent cycles of fasting, either 48 or 60 hours every other week. The fasting alone, with zero pharmaceutical intervention, reduced metastatic tumor spread by up to 58% in certain cancer types and extended survival significantly. The mechanism was that fasting starved cancer cells while boosting the immune system's ability to identify and eliminate them. A subsequent study published in Cell Stem Cell revealed the stem cell regeneration mechanism. When mice fasted for 2 to 4 days, their white blood cell counts initially dropped, which sounds bad but is actually part of the adaptive response. The body was clearing out old damaged immune cells. Then during the refeeding phase after the fast, there was an explosion of new immune cell production driven by stem cell activation in the bone marrow. The fasting essentially forced the body to recycle old immune components and rebuild the immune system with fresh cells. The key pathway involved is called IGF-1, insulin-like growth factor 1. In the fed state, IGF-1 levels are high, which promotes growth but suppresses stem cell activation. During fasting, IGF-1 levels plummet by up to 70%. This IGF-1 decline is the signal that tells stem cells in bone marrow to activate and begin regenerating immune cell populations. Think of high IGF-1 like a brake on stem cell function. Fasting releases that brake. Additionally, fasting activates a cellular recycling process called autophagy, which literally means self-eating. During autophagy, cells break down damaged proteins, dysfunctional organelles, and cellular debris, then recycle the components to build new functional cellular structures. It's like your cells conducting a deep cleaning and renovation project. Autophagy declines with aging and in disease states. Fasting is one of the most powerful known triggers of autophagy in humans. A study from the Massachusetts Institute of Technology demonstrated that even a 24-hour fast doubles autophagic activity in intestinal stem cells, improving their regenerative capacity. Longer fasts of 3 to 5 days produce even more profound effects throughout multiple tissue types. Now, all of this animal research is compelling, but the critical question is whether these mechanisms operate in humans. Multiple human trials have now confirmed they do. A clinical trial published in Cancer Research examined cancer patients undergoing chemotherapy who voluntarily fasted for 24 to 72 hours before and after their chemo treatments. Patients who fasted reported significantly reduced side effects, including less nausea, less fatigue, less weakness, and better maintenance of quality of life compared to patients who ate normally through treatment. More remarkably, some patients experienced better tumor responses, meaning the cancer shrank more effectively with fasting plus chemo than with chemo alone. The fasting was simultaneously protecting normal tissue and sensitizing cancer cells to treatment. This is exactly what the animal studies predicted. A landmark human trial published in Science Translational Medicine tested a 5-day fasting mimicking diet, not complete water fasting, but a very low-calorie, low-protein, low-carbohydrate protocol designed to trigger the same metabolic shifts as fasting while providing minimal nutrition for safety. 100 healthy adults completed 3 monthly cycles of this 5-day protocol. The results were remarkable. Body weight decreased as expected, but more importantly, visceral fat, the dangerous inflammatory fat around organs, decreased significantly while lean body mass was preserved. Fasting blood glucose dropped by an average of 12 milligrams per deciliter. IGF-1 levels dropped dramatically during the fast, then returned to lower than baseline levels after refeeding, suggesting a beneficial reset of growth signaling. Inflammatory markers, including C-reactive protein, decreased significantly, and stem cell markers increased, indicating activation of regenerative pathways. A subset of participants who started with elevated disease risk markers, including high blood pressure, high cholesterol, or high fasting glucose, showed even more dramatic benefits. Their metabolic health markers improved substantially, effectively reversing pre-disease states. So what exactly is happening during these 5 days at the cellular and molecular level? Let me walk you through the timeline of biological changes, because understanding the progression helps you appreciate why 5 days is the optimal duration. Not 3 days, not 7 days, but specifically 5. Day 1, glycogen depletion and metabolic transition. During the first 24 hours without food, your body burns through stored glycogen in your liver and muscles. Glycogen is your short-term energy storage, essentially chains of glucose molecules. You have enough glycogen to fuel approximately 18 to 24 hours of normal activity. During this first day, your blood glucose remains relatively stable because your liver releases glucose from glycogen breakdown. Insulin levels begin dropping significantly since you're not consuming carbohydrates. This insulin decline is the first signal that growth pathways are shutting down. Day 2, ketogenesis activation and growth pathway suppression. By the second day, glycogen stores are depleted and your metabolism must shift fuel sources. Your liver begins breaking down fatty acids and converting them into ketone bodies, including beta-hydroxybutyrate and acetoacetate. These ketones become your primary fuel source, particularly for your brain, which cannot use fatty acids directly. This metabolic state is called ketosis. Ketone levels rise progressively throughout days 2 through 5. Typically reaching 3 to 5 millimolar by day 4 or 5. Simultaneously, IGF-1 levels drop significantly, often by 50% or more by day 2. This IGF-1 decline signals cells to shut down growth pathways, including the mTOR pathway, mechanistic target of rapamycin, which is a master regulator of cell growth and division. When mTOR is suppressed, cells shift from growth mode to maintenance and repair mode. This is when differential stress resistance begins. Normal cells adapt to low mTOR by entering protective states. Cancer cells cannot adapt effectively and begin experiencing stress. Day 3, autophagy activation and cellular cleanup. By day 3, autophagy is fully activated throughout your body. Cells are now aggressively breaking down damaged components and recycling them. Old dysfunctional mitochondria get removed through a process called mitophagy, mitochondrial autophagy. Misfolded proteins that can cause cellular dysfunction get degraded. Damaged cellular membranes get recycled. This is deep cellular renovation happening simultaneously across all tissue types. Your immune system also begins clearing out old senescent immune cells. Senescent cells are cells that have stopped dividing but remain metabolically active, secreting inflammatory compounds that damage surrounding tissue. They accumulate with aging and disease. Fasting triggers apoptosis, programmed cell death, in many of these senescent cells, effectively clearing them from your system. Day 4, stem cell activation initiation. By day 4, the combination of low IGF-1, low insulin, suppressed mTOR, elevated ketones, and cleared senescent cells creates the precise conditions for stem cell activation. Stem cells in your bone marrow, which have been quiescent, receive molecular signals to begin preparing for division. The specific signals include elevated ketone bodies, which act as signaling molecules beyond just fuel, reduced inflammatory cytokines, which normally suppress stem cells, elevated glucocorticoids from your adrenal glands, which prime stem cell niches, and expression of stem cell mobilization factors. However, the stem cells don't fully activate yet during the fast. They're priming for activation that will occur during refeeding. This is a critical point. The magic happens not just during the fast, but during the transition from fasting to refeeding. Day 5, maximum metabolic adaptation and preparation for regeneration. By day 5, you've reached maximum metabolic adaptation. Ketone levels are at their peak. Autophagy is at maximum activity. IGF-1 and insulin are at their lowest. Growth pathways are maximally suppressed, and stem cells are primed and waiting for the refeeding signal that will trigger their proliferation. This is also when cancer cells, if present, are experiencing maximum metabolic stress. They've been deprived of glucose for 5 days. They cannot effectively utilize ketones the way normal cells can. Their growth pathways are still trying to function, but without adequate resources. They've accumulated significant oxidative stress, and they've become sensitized to any additional stress, whether from immune system attack or therapeutic intervention. Then comes the critical refeeding phase. When you break the fast and begin eating again, several things happen rapidly. Insulin levels rise moderately. IGF-1 levels begin recovering. And the stem cells that were primed and waiting receive the signal that resources are available again. This triggers a burst of stem cell proliferation and differentiation. New immune cells flood out of bone marrow. Damaged tissues throughout your body receive stem cells for repair. And your body essentially regenerates itself using the cellular cleanup that occurred during the fast as raw materials. A study from the University of Southern California measured immune cell populations before, during, and after a 3-cycle fasting mimicking protocol in humans. During the fast, white blood cell counts dropped by approximately 30%. But after refeeding and recovery, white blood cell counts rebounded to levels 20% higher than baseline, and the cellular composition showed a higher proportion of naive immune cells, meaning fresh new immune cells rather than old exhausted ones. The immune system had been rebuilt. Now let me address the cancer starvation mechanism more specifically, because this is critical for understanding why this works. Cancer cells have a fundamentally broken metabolism called the Warburg effect. Normal cells, when oxygen is available, fully oxidize glucose in mitochondria through a process called oxidative phosphorylation, which is very efficient and generates 36 molecules of ATP per glucose molecule. Cancer cells, even when oxygen is available, preferentially ferment glucose to lactate through glycolysis, which only generates 2 ATP per glucose molecule. This sounds stupid. Why would cancer cells use such an inefficient process? Because the glycolysis pathway, while inefficient for energy, generates enormous amounts of biosynthetic intermediates that cancer cells need for rapid division. Glycolysis produces nucleotides for DNA synthesis, amino acids for protein synthesis, and fatty acids for membrane synthesis. Cancer cells sacrifice energy efficiency for growth capacity. But this creates a critical vulnerability. Cancer cells become addicted to glucose and cannot easily switch to alternative fuels. When you fast and glucose becomes scarce while ketones become abundant, normal cells switch to using ketones and fatty acids efficiently. Cancer cells struggle because their mitochondria are damaged and cannot effectively metabolize ketones. They're essentially starving even though your body has plenty of stored energy in the form of fat. Additionally, cancer cells require constant growth signals from insulin and IGF-1 to maintain their proliferative drive. When these hormones plummet during fasting, cancer cells lose critical growth signals while simultaneously being deprived of their preferred fuel. This combination creates severe metabolic stress that can trigger apoptosis or make cancer cells vulnerable to immune system elimination. A groundbreaking study published in Nature examined what happens to cancer cells during fasting at the molecular level. Researchers tracked cancer cell metabolism using radioactive tracers that allowed them to see which fuels cancer cells were using. During fed conditions, cancer cells consumed glucose voraciously. During fasting conditions, glucose availability plummeted, and while normal cells switched to fat metabolism, cancer cells showed metabolic chaos. They attempted to use amino acids, they tried to cannibalize their own proteins, and they accumulated toxic metabolic byproducts. Many cancer cells underwent apoptosis within 72 to 96 hours of nutrient deprivation. Critically, when researchers looked at cancer stem cells, the subpopulation of cancer cells that drive recurrence and treatment resistance, these cells were even more vulnerable to fasting than bulk cancer cells. Cancer stem cells are typically more resistant to chemotherapy and radiation than regular cancer cells, but they were more sensitive to metabolic stress from fasting. This suggests fasting could target the exact cells that conventional treatment struggles to kill. Let me be extremely clear about something before we get to the protocol. This is not a cancer cure. This is not a replacement for medical treatment if you have been diagnosed with cancer. What the research shows is that short-term fasting creates metabolic conditions that simultaneously starve cancer cells while regenerating healthy immune and stem cell populations. For cancer prevention, this represents a powerful tool. For cancer treatment support, this should only be done under medical supervision as an adjunct to conventional therapy, not a replacement. Here is the exact 5-day protocol based on the clinical research. This is called a fasting mimicking diet, not complete water fasting, because complete extended water fasting carries risks that require medical supervision. The fasting mimicking diet provides minimal nutrition, approximately 700 to 1100 calories per day, designed to trigger the same metabolic shifts as complete fasting while maintaining safety. Day 1 protocol. You consume approximately 1100 calories, consisting of small amounts of vegetable-based soups, herbal teas, small portions of nuts, and lots of water. The macronutrient breakdown is approximately 10% protein, which is critical because protein restriction is necessary to suppress IGF-1, 56% fat, primarily from nuts, avocados, and olives, which provide satiety without spiking insulin, and 34% carbohydrates from non-starchy vegetables. You're eating, but barely. The point is to provide just enough nutrition to make the protocol sustainable while triggering the fasting response. Most people find day 1 relatively easy because they're not yet adapted to the low-calorie state. Days 2 through 5 protocol. Calories drop to approximately 700 to 800 per day with the same macronutrient distribution. Meals consist primarily of vegetable broths, small amounts of nuts, herbal teas, and water. The food volume is small enough to fit in 2 or 3 small meals or snacks spread throughout the day. By day 2 or 3, most people experience hunger that then paradoxically decreases by day 4 as ketones rise and appetite-suppressing effects of ketosis kick in. Energy levels often dip on days 2 and 3 as your metabolism transitions, then rebound on days 4 and 5 as you become fully keto adapted. Throughout all 5 days, you must maintain adequate hydration. Drink at least 2 to 3 liters of water daily. You can include herbal teas, black coffee, or green tea without sweeteners. Adequate hydration is non-negotiable during fasting protocols. Day 6 and beyond, the refeeding phase. This is as important as the fasting phase itself. You cannot go from 5 days of severe calorie restriction directly back to your normal diet without causing digestive distress and potentially dangerous refeeding syndrome in extreme cases. Break the fast gradually over 2 to 3 days. Day 6, consume approximately 1200 to 1400 calories focusing on easily digestible foods, including cooked vegetables, small amounts of fruit, vegetable soups with added healthy fats, and potentially small amounts of fish or eggs if you consume animal products. Avoid large meals. Eat small portions every few hours. Day 7. Increase to approximately 1600 to 1800 calories with similar food choices, adding more protein and complex carbohydrates from whole food sources. Day 8. Return to your normal healthy eating pattern, ideally focusing on whole foods, abundant vegetables, quality proteins, and healthy fats, while minimizing processed foods and refined sugars. The research protocols typically involve doing this 5-day cycle once per month for 3 to 6 consecutive months, then potentially quarterly for maintenance. The monthly frequency allows your body to undergo repeated cycles of cellular cleanup and regeneration without the risks associated with chronic calorie restriction. Now, from clinical practice and research protocols, here are 6 critical optimization details. First, prepare your body before starting. If you currently eat a standard American diet high in carbohydrates and processed foods, going directly into a fasting mimicking protocol will be miserable. Spend 1 to 2 weeks before your planned fast transitioning to a lower carbohydrate, whole food diet. This pre-adaptation makes the metabolic transition to ketosis much smoother. Second, plan the 5 days strategically. Do not attempt this during a week with major work deadlines, intense physical training, or significant stress. Choose a week where you can prioritize rest and have flexibility in your schedule. Many people find starting on a Monday and finishing on Friday works well, allowing weekend recovery. Third, monitor your response objectively. If possible, measure ketone levels using blood ketone meters starting on day 3. You're looking for blood ketone levels between 1 and 5 millimolar, which confirms you're in therapeutic ketosis. You can also track fasting blood glucose, which should drop into the 70 to 90 milligram per deciliter range by days 4 and 5. Fourth, modify the protocol based on your metabolic health status. If you're already lean and metabolically healthy, the standard protocol works well. If you're overweight with insulin resistance or type 2 diabetes, you may need medical supervision and potentially modifications. If you're underweight or have a history of eating disorders, fasting protocols are not appropriate. Fifth, consider the timing relative to cancer treatment if you're using this as a treatment adjunct. The research suggests fasting for 24 to 48 hours before chemotherapy and continuing for 24 hours after provides maximum protection to normal cells while sensitizing cancer cells. This requires coordination with your oncologist. Sixth, support the protocol with appropriate supplementation. During the 5-day fast, take a multivitamin to prevent micronutrient deficiency. Take electrolytes including sodium, potassium, and magnesium, especially if you're experiencing headaches, muscle cramps, or fatigue. Consider omega-3 supplementation to support anti-inflammatory effects, and potentially use exogenous ketones on days 3 through 5 to ease the transition into ketosis and reduce symptoms of keto flu. Let me tell you the 6 mistakes that sabotage this protocol and create unnecessary suffering or prevent results. Mistake 1, not restricting protein sufficiently. The IGF-1 suppression that's critical for stem cell activation requires protein restriction to approximately 10 to 15 grams per day during the fast. If you're consuming significant protein, you won't achieve the metabolic shift. This is why the protocol emphasizes vegetable-based foods and minimal nuts rather than protein-rich foods.